Hello, all from balmy Central Iowa. Well, Central Iowa. And welcome to the virtual Harmful Algal Bloom Research Symposium. My name is Rick Cruz. I work at Iowa State University and am director of the Iowa Water Center. I'll be the moderator for this session. The symposium is hosted by the Algal Bloom Action Team, of which I am a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the north central region of the U.S. Team members include National Network of Water Resource Research Institutes, the North Central Region Water Network, and University Extension within each state in the north central region. Today is the third session of the symposium and will focus on human health and social well-being and HABs. It will last 2 hours and feature 3 presentations on emergent harmful algal blooms or HABs research from leading researchers across the north central region. We'll also have a 10-minute break at 9:45. Following today's presentation, we'll also host a 1-hour facilitated discussion on the topic of today's theme, to discuss how we can better disseminate information regarding HABs. This discussion will help the work of this team, but due to the number of people at the symposium and the platform constraints in this virtual reality, an additional sign-up was required for the facilitated discussion, and all the spots have been filled. We apologize for any inconvenience that if you were not able to sign up and you wish to. We've been blown away literally by the interest in today's event, but we'll make sure that we can accommodate additional folks at future events. Our team is working to develop a website of resources, including fact sheets, FAQs on HABs topics. Additionally, we plan on organizing webinars, recording video content, and to continue to host this symposium in the future. However, to do that, we're seeking your insight, your input on how we can improve the dissemination of these materials and information in content areas of research for the public. If you have any questions about the facilitated discussion, or if you are having any technical issues, please note that in the chat box, and our team will do the best to assist you. If you have questions about questions for the presenters, also please place them in the Q&A panel, and I will ask them of our presenters today as time allows following their presentations. Before we get started, I'd like to take a moment and thank our sponsors for making the symposium possible. In addition to the support of the North Central Region Water Network and the National Network of Water Resources Research Institutes, we want to thank our champion sponsors, FlowCam by Yokogawa Fluid Imaging Technologies, Limotech for their support. All right, let's get started. Today's topic, as mentioned, is human health, social well-being, and HABs. And we have 3 great speakers lined up for you today. Our first speaker is Lesley D'Anglada, Senior Microbiologist at the Office of Senior Technology, Office of Water, US EPA. Most of you can see this panel. My half of this is hid on my screen. Technology is marvelous when it works. Lesley, we contacted EPA, was happy to join us. She has an excellent set of credentials, as you see in front of you. And at this time, Lesley, we really appreciate you joining us, and the floor is all yours. I'm going to stop sharing my screen, and you go ahead and share yours.
Thank you, Rick. All right. Good morning. I hope you can see me. And I hope you can see my presentation.
We can see your screen.
Okay, let's try again. Right here. Can you see the presentation now?
Yes, we can.
Wonderful.
Lesley, we do see your notes as well.
All
Yes.
right. So, sorry about that.
No problem. We've all lived in a virtual world and we're still learning how to do it right.
I know. I keep doing the same mistake. My apologies. I'm gonna get this. Promise. Promise. And then go here. There you go. Can you see my presentation now?
Perfect.
Wonderful. Yeah, I got it. Okay, good morning. Thank you for the introduction and for the opportunity to present today on the risk assessments that we did for 3 cyanotoxins that we can find in drinking and recreational waters in the US. So I will start this presentation with summarizing the adverse health effects from exposure to 3 common cyanotoxins in the US. And then I will provide an overview of EPA's approach to deal with harmful algal blooms in freshwater systems. In freshwater systems, cyanobacteria are the most common type of algal bloom, although they are bacteria and not properly algae. There were presentations yesterday that discussed the frequency of HABs in the US, and there was some discussion about it. What we know is that we have not only seen an increase in occurrence, but also a combination of the increased awareness, monitoring, and reporting. We have seen several major cyanobacteria harmful algal blooms events that had been reported in the past few years. In this slide, I'm introducing the One Health concept, as HABs are a good example of a One Health problem. HABs could affect people, could affect animals, and could affect our environment. We heard about that yesterday, and requires an interdisciplinary approach to achieve optimal health outcomes. The effects of harmful algal blooms on people, animals, and the environment also have economic consequences. And I believe that our next presenter will be talking about those economic impacts. Illnesses from exposure to cyanotoxins vary depending on the toxin you are exposed to and the dose of the toxins you are exposed to. There are several ways people can be exposed to cyanobacterial toxins. You can be exposed through the skin, to cyanobacteria cells and also the toxins while recreating in toxin contaminated waters. You can breathe toxins in aerosols. We know that cyanotoxins, they don't evaporate, so they're not gas, but they could be found in tiny airborne droplets or mists with toxins, either in recreational areas or by wind-blown spray. You can ingest cyanobacteria and their toxins incidentally while you are recreating, or you can ingest cyanotoxins in drinking water or contaminated food. Another example of the route of exposure is via intravenous exposure, for example, via dialysis. That has been the only recorded death from exposure to cyanotoxins. In this case was Microsystems in a dialysis clinic in Brazil a few years ago. But that method of exposure is different than ingestion. It was directly in through the circulatory system. We don't have adequate toxicity data for some routes of exposure to determine the health risk from exposure to cyanotoxins, but we do have data for some toxins from exposure to cyanotoxins through the oral route, which is the ingestion in drinking and recreational waters. And that is the toxicity data that I will be discussing today. But before I discuss that, let me briefly summarize how we develop these risk assessments and how I prepared these slides. We work along with Health Canada, started back in 2012, to develop guidance values in drinking water for 3 cyanotoxins: Microsystems, Anatoxin A, and Cylindrospermoxin. At that time, we used the 2006 draft toxicological reviews that the Office of Research and Development of EPA developed. We updated them, and then we used them to develop what we know as a health effects report document, which is a comprehensive review of the health effects in which we include the quantification of the dose response and the derivation of the reference dose for that cyanotoxins. We derive a reference dose to be used to develop guidance values, and we evaluate dose response data from case reports, which are epi studies or human case poisonings that have some or may have dose response data, which in most of the cases, we don't have that data. So we use laboratory studies, laboratory toxicity studies using mice and rats to develop numeric toxicity thresholds that are applicable to humans. So we put that in a formula and we add some uncertainty factors and we come up with a guidance value. We use different types of studies. We evaluate acute exposure, which is usually a study that is 24 hours. So it's that acute to high levels of toxins that you can be exposed to. We develop short-term subchronic studies, which is that exposure that you can be from our studies from 30 to 90 days. So that's a very short-term kind of 2 weeks exposure. And then we do a long-term exposure. What happens if you are exposed to these toxins for a long period of time? And we use chronic studies that are usually the period of the study is more than 90 days. So in the following slides, you will see that I discuss the summary of the health effects that we found based on these types of studies: acute studies, subchronic short-term studies, and chronic studies. In addition to that, we also did an evaluation on the possibility of carcinogenicity of these toxins, and it's included in this presentation. So we start with microsystems. I know you heard a lot about microsystem yesterday. Microsystem is a small cyclic peptides with 5 amino acids. The common variations of microsystem involve the substitution of 2 amino acids in the 2nd and 4th position of the peptide. And this makes the formation of hundreds of microsystem variants. So we know about more than hundreds toxin variants or congeners. Being microsystem LR, the most studied microsystem variant. Most of the toxicological database on the effects of microsystem is limited to the LR congener. Therefore, the evaluation of health effects from exposure to microsystems is based on studies evaluating the effects produced by microsystem LR. That doesn't mean that there are other congeners that are not toxic or that are not more toxic. It means that the toxicity data that is available is based on that particular congener and the one that we can use. So when microsystem is ingested, it's transported from the intestinal tract throughout the body in the circulatory system. Once it reaches the liver, microsystem has high affinity for the cytosolic proteins in the liver, the protein phosphatases 1 and 2, PP1, PP2, which result in their retention in the liver. So when it passes the liver, they see these enzymes immediately do a covalent bond, which is a very strong bond, and they start inhibiting enzyme activities of the PP1 and PP2, resulting in what is known as liver toxicity. The limited data in humans are mostly from the accidental ingestion of toxin-contaminated water during recreational activities. And these reports demonstrate the exposure to microsystem could affect the liver. However, these human data or case reports couldn't be used for the derivation of a reference dose for microsystems, as they are limited by the lack of toxins data, as one of them didn't report its cyanotoxins concentrations and the possibility of co-exposure with other microorganisms. One of the studies, they developed GI illnesses. There were microsystems levels, but also there were high levels of E. coli in the water that could have done some of the GI effects. Another was a person in Argentina who drank water and had Same kind of GI effects, develop pneumonia, and at the end, the enzyme levels of the liver were really high. He recovered from that exposure in 20 days. But there were no concentrations or dose response on what was the concentration of the toxins in the water when he was exposed to it. Laboratory studies of microcystin LR have found adverse effects on the liver and also on the reproductive system. Liver effects are the health effects more common from exposure to microcystin and are shown as an increase in liver weight, bleeding in the liver, and an increase in liver enzymes in the blood. Reproductive effects were reported by a single study in 2011, showing effects in ovaries and testes, the decrease in sperm counts, and found and reduction in sperm motility. We evaluated that study, and we found limitations in the design of the study, in the reporting of the data, and also on the methods they used to analyze the data. After the development of the risk assessment for microcystin in 2015, new studies of the toxicity of microcystin have been published. However, they find some adverse effects in the female and male reproductive systems, but they have not been able to determine what is the mechanisms for the production of those eggs, for the manifestation of those endpoints. And therefore, we believe that that relationship needs to be further researched. There are two chronic studies that have not observed clinical signs of toxicity. Both are limited by the design and data reported. One study was done over 18 months and used only one dose and find no toxicity. And the other one was done for more than 200 days. And the quantitative data on the toxin was not reported. So we still don't know what are the chronic effects from exposure to microcystins. The next toxin is Cylindrospermoxin. We know two naturally occurring congeners and two analogs from the Thai strain of what was previously known as Cylindrospermopsis raciborski. Cylindrospermoxin may be retained within the cell, and I know we talked a little bit about that yesterday when we talk about treatment. But half of the time and half of the toxin is released to the water without breaking the cell, without cell lysis, and is found dissolved or attached to other materials in water. Although the mechanism of the toxicity of Cylindrospermoxin has not been fully characterized, we know that once ingested, Cylindrospermoxin is absorbed from the gastrointestinal tract to the blood and is circulated through the body. Once it gets to the liver, very similar to microcystins, Cylindrospermoxin binds to the cytochrome P450 enzyme and inhibit the hepatic protein synthesis, affecting both the liver and kidneys. Even though they are both hepatotoxins, meaning that they affect the liver, the mechanism of toxicity for Cylindrospermoxin, we believe is different than the mechanism for of toxicity of microcystins. Human data on the toxicity of Cylindrospermoxin is from an outbreak in Australia caused by a short-term consumption of drinking water contaminated with Cylindrospermoxin raciborski. The major adverse effect observed was kidney damage. Data on Cylindrospermoxin was not reported, and there were very high concentrations of copper sulfate in the water. Therefore, there are some confounding factors that we could not, that influence the observed symptoms. Acute and subchronic animal studies have found kidney effects manifested as an increase in kidney weight, decrease in protein creatinine ratio due to glomerular dysfunction, which means that there are more creatinine in the blood than proteins, which is normal in the urine. I'm sorry, and also liver effects that were manifested by lipid infiltration and increase in liver weight. The liver effects that were observed in these studies were only on the highest doses, which means that kidney is the most sensitive organ for Cylindrospermoxin damage. we have not seen any chronic studies done for Cylindrospermoxin. The last toxin I will be discussing today is Anatoxin A. Anatoxin A has one known analog and 4 derivatives. Although the toxicity data on this derivative is limited, derivatives of anatoxins bind to the same receptor and may have similar potency to Anatoxin A when administered orally. So we estimate that they will behave the same way that Anatoxin A behaves. Anatoxin A has been associated to the death of livestock and dogs. However, linking that exposure to the effect is usually difficult because Anatoxin A undergoes rapid degradation in the presence of sunlight, which makes it hard to detect during an animal poisoning events. So when you have the animal and it goes to the veterinary and then they go back, not the veterinary, but the state or the agency or the administrator of the lake goes and takes samples, Anatoxin A may not be present in the water. So it's hard to make that relationship. We know that once Anatoxin A is ingested and distributed in the blood, Anatoxin A attached to neuromuscular nicotinic receptors located in the brain, and it starts a physiological response in the central nervous system. So Anatoxin A is a neurotoxin. Once Anatoxin A binds to the receptors, it stimulates the muscle cells, causing fatigue, muscular twitching, and paralysis. So that overstimulation tends to stimulate the respiratory muscles as well, which could cause respiratory arrest, and that causes a very rapid death. There are no human data, thank God, of oral toxicity of anatoxins. There's one single dose response study found that deaths occur within 2 minutes of administration of Anatoxin A, which tells us that Anatoxin A has a high acute oral toxicity. The animals that pass away show loss of coordination, muscular twitching, and death by respiratory paralysis. There are 2 short-term subchronic studies. The studies are limited because they evaluated a few endpoints, and some of the endpoints that were evaluated were not related to neurotoxicity. So they evaluated changes in blood and other endpoints that are not related. So we don't know what was the effect. They provided limited quantitative data and used partially purified extracts of the toxin. One study found several minor hematology and blood chemistry changes that were not considered to be toxicological significant, and reported a death of a single mouse that couldn't be explained. In the second study, the highest dose did not cause any adverse effect. We don't have any chronic studies for Anatoxin A. We also evaluated the carcinogenic potential of these 3 cyanotoxins, and I think later today you will hear a little bit more about carcinogenic effects on microcystin. For microcystin, several human EPI studies have reported an association between microcystin in drinking water and the occurrence of liver and colon cancer. These studies have some limitations. For example, one of them was an ecological study, which is a type of study that is not able to prove cause and effect association. Other studies have poor measures of exposure, potential co-exposure to microbial and or chemical contaminants, and also in some cases they failure to control for non-liver and colorectal risk factors like hepatitis and other factors. Animal studies have suggested that microcystin is a tumor promoter. So microcystin is classified as a group 2B, which is a possible carcinogenic to humans by the International Agency for Research on Cancer. But the limitations in the database of not having adequate long-term dose response studies doesn't allow us to determine the carcinogenicity of microcystins. Several lab studies also have found the potential damage to DNA by Cylindrospermoxin. Again, up to date, we don't have a bioassay, we don't have a long-term study that evaluate the cancer potential of cylindrospermoxin. Dose response data or studies longer than 2 years with more than one species, with multiple doses, and what we call gold standards studies. And those are studies that evaluate different endpoints. Therefore, the currently data is inadequate for us to determine the carcinogenicity of cylindrospermoxin. And for Anatoxin A, there's no data on carcinogenicity. Another exposure we evaluated was the accidental oral ingestion of cyanobacteria cells during recreational activities. We know from EPI studies that the association between cyanobacteria and human health effects is there and includes dermal and allergenic reactions. However, when we evaluated these EPI studies, we observed that the relationship between cell density exposure and specific health outcomes, such as GI effects or dermal symptoms or allergic type of reactions, eye ear irritations, respiratory symptoms, were not consistent. So we observed studies with 5,000 cells per milliliter, and we observed the same effects of someone that was exposed to 100,000 cells per milliliter. So there was a lot of inconsistency in the effects and the cell density. In other studies, we found that associations with different specific health effects that were not related to what we were expecting from exposure to cyanobacteria cells. So as a summary, the adverse effects from exposure to microsystems and cylindrospermoxin are liver toxicity and also kidney toxicity for cylindrospermoxin. The main toxic effect of Anatoxin A is acute neurotoxicity. Unfortunately, based on the limited toxicity data, EPA was not able to derive a reference dose for Anatoxin A and develop a health advisory. We did develop a health effect support document, and we will reevaluate the data to determine if provisional could be developed or if further analysis of the toxicity needs to be done. Exposure to cyanobacteria cells during recreational activities in water contaminated with the cyanobacteria bloom can cause inflammatory effects such as skin, eye, ear irritation, fever, GI illnesses, respiratory symptoms. But at this time, EPA understand that the available data is insufficient to develop a threshold or a quantitative recreational values for total cyanobacteria cells density that are related to inflammatory health endpoints. Several research needs have been identified during the development of our risk assessment. I listed some here. Very important ones for my work are the inhalation, what is the inhalation, and dermal effects from exposure to these cyanotoxins. Another important one is what are the health risks from exposure to a mixture of cyanotoxins? And we saw yesterday from USGS and others that these cyanotoxins are not isolated. They are, there's a mixture of them, and we can be exposed to several of them at the same time. Very briefly, I included here other adverse effects from exposure to cyanotoxins. I will not go into much detail as there will be 2 presentations in the afternoon on the effects of livestock and aquatic animals. But we know that cyanotoxins have been associated with the death of wildlife and domestic animals, not only Anatoxin A, but also other toxins. Animals are either exposed by drinking or by eating or licking their paws, and we have seen a rapid onset of symptoms and sometimes deaths. Fish could also be exposed to cyanotoxins through the food web or directly feed on the ceston or through dissolved toxins in the water. From the review of the literature that we have done, we found that different species have different uptake routes and metabolisms of microsystems specifically. There's a lot of studies on microsystems and less for the other toxins. And we also learned that cyanotoxins can bioaccumulate in fish. and in tissues, although it tends to accumulate more in the liver and hepatopancreas. And we know why, because of the affinity of the toxin. Cyanobacteria cells and toxins have also been or could be found in irrigation water, and they can contaminate plants. They can be absorbed by the roots and gets all the way to the plants and grains. We have observed that the root tends to accumulate more than the leaves. And so at EPA, we are addressing the HABs issue, trying to follow the One Health approach. And these are the different actions that we have used. And I'm going to go very, very briefly on each of them in the next slide. So we develop regulations, guidelines, and recommendations. Currently, cyanotoxins are not regulated under the Safe Drinking Water Act or Clean Water Act. We published in 2015 drinking water health advisories for microcystin and cylindrospermoxin. Health advisories are non-regulatory estimates of a contaminant, which no adverse health effects are likely to occur over a specific period of time. Our health advisories are 10 days health advisories, but we have also 1-day health advisory over a lifetime. For cyanotoxins, we developed 10-day health advisories. On May 2019, we released the recommended recreational water quality criteria and swimming advisories for microcystin and cylindrospermoxin. We use the same toxicity data on short-term studies. We are currently evaluating the health risk of oral exposure to Toxins and also nodularis. And we're also evaluating the adverse effects of cyanotoxins in fish and also in plants that are irrigated with toxin-contaminated irrigation water. Along with the health advisories and recreational criteria, we published several documents in support of these guidelines. We develop methods, we publish recommendation documents and other resources on risk communication. EPA conduct assessments as well. We heard yesterday about the NARS. We conducted a monitoring of 10 cyanotoxins in drinking water systems from March 2018 to September 2020 through the Unregulated Contaminant Monitoring Rule, UCMR. And we use that rule to determine what is the occurrence of contaminants that are not regulated in support of the CCL program and determine if a regulation is needed. We, as I mentioned, NARS is the National Aquatic Research Survey. We also conduct monitoring in other aquatic sources like that, like lakes, rivers, streams, ocean and coastal waters and wetlands. Microsystems, Toxicity, Cylindrospermoxin were included in the 2007 National Lakes Assessment, were included in 2012, and EPA hopes to keep including cyanotoxins in future surveys. We do a lot of outreach and communications. We keep the Cyanobacteria HABs website. We conduct national webinars. For the past 5 years, we have been doing regional workshops across the US on harmful algal blooms in 9 of the 10 regions of EPA regions across the US. We have partnerships and collaborations. We participate in several federal and non-federal work groups, and our regional offices also collaborate with states, agencies, and universities. And lastly, we do research. EPA's research is mostly done by the Office of Research and Development, but also the Office of Water, Office of Science and Technology, as well as the Office of Groundwater and Drinking Water and the regions. We also conduct our own research in partnership, sometimes with either the states or universities. Here's my contact information. I wanted to show you this beautiful dog. Her name is Pandora. And she was one of 6 dogs that in the summer of 2018 were exposed to a microcystis bloom in Martin County in Florida. Although very sick, thanks to the prompt veterinary care, 5 of the 6 dogs survived, including Pandora. And since then, she has been actively advocating for clean water in Florida. So we need to support her. And the story of these 6 dogs was published in 2019 in the journal Toxins, and I show here a little bit of the journal right there. And it's important that we also communicate the risk to veterinary, to other medical professions. So prompt attention to when they are exposed is done. And so I think CDC is doing a great job in promoting that outreach and communication of risk. That's it for me. Thank you for your attention.
Hey, thank you, Lesley. Thank you. Really, really appreciate it. And as a member of the planning committee, you hit the target. You hit bullseye. Absolutely perfect.
Oh,
we have a series of questions, and let me read the questions to you one at a time. And if you can respond to those questions, we'd be greatly appreciative, very appreciative.
of course.
And the first one that I have on my list is, as you pointed out, it is difficult to ascertain exposure post facto due to the transient nature of toxins in the environment. Can you address the status of clinical diagnostic tests under development that can confirm exposure through urine, hair, fur, etc.?
That's a great question. And I know that the Centers for Disease Control, CDC, is developing these biological methods to detect cyanotoxins in blood and in urine. I don't know the status of it. I know that they're in process, but I can put you in contact with the person that is developing those methods.
Okay, thank you, thank you. The next question, are HABs not included in the Clean Water Act because they are a product of industrial agriculture?
Well, no. HABs are naturally occurring events that are promoted by certain factors, and all this was discussed yesterday, and one of them is an excess of nutrients that could happen from agriculture, right? So we have several programs under the Clean Water Act in order to control the release of that excess of nutrients and the source water protection. So even though, since they are a natural occurring event, they are not explicitly included in the Clean Water Act. The different actions under the programs of the Clean Water Act do address harmful algal blooms.
Okay, thank you. Will there be guidelines developed for concentrations in fish, shellfish, and in plants?
Very good question, Kathy. You know, we collected the information, and the information from the irrigation water was an initiative from Utah. So I really appreciate their concern and reaching out to us to address this issue. From the information that I have evaluated, we don't have enough data on those response and toxicity effects to develop guidelines, values, or thresholds for fish, shellfish, and in plants, or plants or irrigation water. I think what we want to do is to do a comprehensive literature review, bring all that information together, provide what are the risks, and maybe provide some recommendations on how to avoid exposure. But I understand that from what I've seen, I don't think we have that data yet.
Mm-hmm. Can you provide the reference for the reproductive effects of MCLR that are credible?
I can provide the references from the studies that have been done, and I think you choose if they're credible or not, but I can provide the references of the different studies, yes.
Is cell density adverse effects an area of current research?
Cell density is always an area of current research. Yes, it is. I think we have, in different ways, we are evaluating how cell density could be related to microsystems, and it's related using different models. Like, for example, the draft lake criteria is based on that, how cell density could be related to microsystem concentrations. So I envision that we will continue working on that.
Man, Lesley, I like your answers. They're short and to the point. You get to more questions. NTP has listed MCLR for chronic testing. Will this happen?
We were in contact with them in 2015, and it wasn't for chronic testing, but it was for repro development. And then priorities changed, and I haven't heard from them again. If you have a contact that I can reach out to, just pass it on to me and I'll follow up. But to my knowledge, I haven't heard anything about that.
From Denise, which cyanobacterial cells is the most dangerous? Microcystin has been studied most, but the antitoxin A seems very acute responses.
Good question, Denise. I think you're referring to cyanobacterial algae, right? Microcystin has been studied most. We could say because it's widespread, but also it's very easy to find, right? And we have methods to develop. We have standards to analyze it. It's an easy toxin. Anything is toxic depending on the dose. So microcystin could be as toxic as Anatoxin A. The mode of action, the mode they affect is what is different. So microcystin could take a longer time to see an effect, while Anatoxin A is a neurotoxin. So you will see the effects immediately. It all depends on the dose. Anatoxin A has been very difficult to study. We don't have, finding the standards to do the studies are expensive and difficult to determine the purity of those. So doing laboratory studies is an expensive process. And so we are working on to do that for Anatoxin A. But since they're different toxins, I don't want to say that one is more toxic than another. But the mode of action of Anatoxin A is more acute. So you will see the effects directly and maybe more intense, depending on the toxicity, on the dose.
What sources did you use to obtain your data? CDC, state records, local health departments?
I'm assuming the data is on the toxicity studies. These are toxicity studies that have been done by either universities, agencies. For example, the one that we use for microcystin LR is from the German Public Health Agency. There's another study that was done by and sponsored by WHO. The one that we use for Cylindrospermoxin is from Australia. If CDC has a study, we evaluate it. And so local health departments also, if they have studies, we evaluate all the data that is published.
Okay, thank you very much. We're bumping up against break time. It's 9:44 plus a whole bunch of seconds. So Lesley, perfect, absolutely appreciate what you've done and how you've done it. So the rest of the troops, for everybody, we have a 10-minute break at this time. Possibly Lesley will have a chance to answer questions that are in the Q&A box. I think she will have access to those. So we'll see you back here at 9:55. Thank you.
Thank you.
Yes, thank you, Lesley. If you're able to stay on for a few minutes and type in some answers to the Q&A, certainly love that. But if you have to leave for another commitment, we certainly understand.
I'll address them now, no worries.
Thank you so much. And Rick, if you would be able to share your screen before having a quick coffee or something like that, that would be great, just to make sure that we are giving our sponsors some visibility as promised.
Yep, absolutely. I will. I need to disconnect first and reconnect to another source, and then I'll share
yes,
screen.
Of course.
Okay, thank you. Dr. Zhang is also affiliated with the Center of Agriculture for Agriculture and Rural Development, CARD. Wendong, Dr. Zhang, it is yours. I'm going to stop sharing my screen, and if you can start sharing yours, the floor is yours.
Great. Thank you. Good morning, everyone. Let me share my screen. Rick, can you see the slides okay?
Yes, I can. Very good.
All right, great. So good morning, everyone. My name is Wendong Zhang from Iowa State University. I actually, I'm a PhD graduate student from Ohio State, and I had a pleasure to work with Dr. Jiyoung Lee, who will speak after me as well. So today I'm going to talk a little bit differently rather than honing on the harmful algal bloom, but talk about the front end and the and also the afterwards effects from both the economics perspective. So mainly I will talk about the economic benefits, and I will also talk about some of the ongoing research related to economic cost on mitigating and preventing harmful algal blooms. Okay, before I jump into the presentation, I want to give you a little bit background about myself. As I said, I got my PhD from Ohio State on agricultural and environmental economics. And importantly, here on Iowa State, I currently lead one of the surveys that try to quantify the economic benefits of water quality, known as the Iowa Lakes Survey. This has been led by Dr. Cassie Kling before she left for Cornell. This current survey has just completed the 2019 series, where the whole series started in 2002. We're about to announce the results probably over the next following months. So with that, today I'm going to talk about some of the recent and ongoing work I'm doing related to conservation practice adoption, harmful algal blooms, and related to cost and benefit analysis, especially related to agricultural conservation policies. As you can see that one of the things I do is I do a lot of surveys of farmers and general public. On one hand, I try to understand what motivates farmers and landowners to adopt water quality improving conservation practices. At the same time, I also do surveys of the general public as well as the recreational users, such as recreational anglers, to try to quantify what is the economic benefits of improved water quality. Okay, so these bolded sections are the ones that I will highlight today. And we actually, as Rick mentioned, we just got a new grant from Iowa Water Center, joint with Iowa Nutrient Research Center, to try to look into the disparate nutrient impacts on lake-based recreation and tourism for socially disadvantaged or underrepresented Iowans. And we are planning to do that by augmenting with our existing Iowa Lake Survey, which tends to undersample these disadvantaged groups. Okay, so I don't need to mention too much of the motivation for this group. In general, when you are looking at my research, I look at two particular regions. One is Lake Erie, where I did my dissertation work, and Iowa is part of the Mississippi River watershed. So I'm also really now increasingly interested in conservation practices that is related to the Gulf of hypoxia in the Mississippi, in the Gulf of Mexico hypoxic zone, right? So when you are looking at these two watersheds, what is interesting is you can see that their water body is different, so the limiting nutrients are different. Where the Gulf of Mexico, in Iowa, the major concern is nitrogen, where in Lake Erie it's a phosphorus. But in this chart on the right, the paper shows that essentially if you want to reduce the hypoxia area significantly, you need dramatic reductions, probably beyond 40 to 50% of the nitrogen reduction in these large watershed, and which is not that different in terms of the have to go when you are thinking about the goal for Lake Erie, right? So Lake Erie, it's thinking about dissolved and total phosphorus, but the goal is also try to do 40% reduction in the Maumee River watershed, which is in the Western Lake Erie Basin, and also the largest watershed for the Great Lakes region, where it's also part of my dissertation efforts. Okay, so broadly, when you are thinking about what are the costs and benefits of mitigating and preventing harmful algal blooms, so Lesley has this morning has talked about one of the really important benefits, which is the potential health benefits, which I would argue that because of the lack of data, lack of systematic data, we actually don't know a whole lot about. And when you are thinking about some of the additional benefits that the economists have better handle on, those are typically includes recreational opportunities and the value of waterfront property values, the increase due to better water quality. There's also research by Dr. Lee, I'm sure she will talk about this later, about how reduction in harmful algal blooms and algal toxins could reduce the wastewater treatment cost and these through the facilities and avoided investments. When you are thinking about the cost, there are two layers that might be meaningful. One is the direct cost for farmers if you impose regulations, or potentially there could be more input cost and increased managed time, and but also when you're thinking about the cost, there's also a cost for taxpayers, which tends to be really important because the current policy is mainly voluntary cost share payments subsidies. That is, when you are thinking about all the USDA programs, that tends to follow essentially rest on the taxpayers, right? So for academic research, we are essentially try to model this in a very interdisciplinary way, where I see that the economists will contribute in the first box, where the economic model will try to quantify under the current and alternative policies how this will induce changes in farmers' land management, conservation practice adoption, and land use changes. And I'm using this Lake Erie as one example, but you can think of this broadly as well. So this land use and economic decision can translate into watershed outcomes. And with help of hydrologists and ecologists, you can turn that into nutrient loadings, either phosphorus or nitrogen, to the desired water bodies. And we also have collaborations with aquatic ecologists who can tell us, for example, if you have 20% reduction in phosphorus loadings, how much more fish population will have, how the water clarity might improve, whether and how frequently we will see the occurrence of harmful algal blooms in these changed ecosystem services. And after that, that's the economists will also come in with the quantified changed ecosystem services. We have models that can quantify and turn those physical terms into dollar terms. And by forming this loop, we can potentially estimate the cost and benefits of different policies, right? I will give you some examples related to that. As I mentioned, when you are thinking about the current existing policies, the blue bar that is rising over time is the mainstay of the agricultural conservation policy, which is a working land program known as the EQIP and CSP, where essentially it's the voluntary subsidy saying that if you adopt cover crops, for example, I will give you $15 or $25 per acre. In terms of subsidy, there's not a whole lot of spatial targeting. There's not much restrictions on who can apply, and there's not a whole lot of targeting in terms of the high runoff sub-watersheds, right? We just have a paper published in Land Economics, which use this integrated economic hydrological model of Western Lake Erie Basin, and here are some of the findings I want to tell you. So here are the 3 BMPs that we focused on in that paper. Again, the key nutrients is phosphorus, right? So we are interested in about subsurface placement of the phosphorus fertilizer to place the fertilizers below the top 2 inches of the soil, cover crops, and also reduction in fertilizer applications. What we find is that if on the left, if you do the current policy, if you use the cost share payments, and if you increase the payments level for these people who are currently not adopting, if you increase the payments level from not paying them to $80 per acre, then you can potentially increase the share of the land in this watershed currently under subsurface placement increase from 47% to closely 63%. So you can achieve, and when you're looking at cover crops in this line, it's even more dramatic increases, which is 20% to 60% increase in terms of the adoption. So when you are paying $80 per acre every year, then you could potentially make sure that half of the watershed is growing cover crops, right? I will go back to the taxes later, but when you are looking at, for example, the impact of the loadings, this is less rosy of the outcome. So just focused on the top 2 figures for now. And this shows you on the left, on the y-axis, it shows you the phosphorus loading due to these adoptions, right? So we have talked with you that if you increase the payments level from $20 to $80, the cover crops and the placement potentially could increase from 45% of the acres to 63%. However, this only increased the loadings from about 2% of the reduction to slightly over 12%. So this is not as proportional as we hope. And when you're looking at one of the magic bullet points lauded in the literature, in the cover crops, we actually find that when you have dramatic increase in cover crops, you have not much increase in the reduction of the loadings. We actually find even when you are increasing the cover crops adoption by 40% of the acreage in the watershed, you only can achieve less than 1% additional reduction in phosphorus loading. There could be multiple factors driving this. And one of the things could be that the effects of the cover crops is uncertain, but also in the models, we have about 2 weeks laid bare after harvest before planting of the cover crops. However, this suggests that if you only look at the adoption acres and say we can achieve a tripling of the cover crops acres, this must translate into dramatic reduction in the phosphorus loadings. You might be wrong, right? So on the right, it shows you another figure that if you use fertilizer taxes, it probably will take you 400 or 300% fertilizer taxes, dramatic tax rate to even see 15% ponds per acre reduction. So this requires drastic changes in the farmers' behavior. One of the nice things in this framework is that it's depending on your perspective, that when you are thinking about your 300% tax, this will impose dramatic costs for the farmers. But we also looked at one of the policies that potentially on the figure D, that we looked into a hybrid policy where you can potentially use the fertilizer taxes to collect revenues and then allocate it back as a share of, as a pool for the funds for cost share payments. When you do that, you can potentially have the double whammy effects of this hybrid policy, and you also avoid some of the criticism that you're taking money away from farmers, but through this, you are essentially redistributing the income to another farmers for the water quality purposes, right? So for the interest of time, I won't go into too much details about the cost, but in general, for this presentation, I will just encourage you to focus on the lower part of the figure, where these are the payments, right? So these are the existing payments for the cost share programs, where when you are spending almost $200 million for the cost share payments in Lake Erie watershed, probably you're only achieving an additional 10% in phosphorus reductions, right? So this one of the key message of this is that you need interdisciplinary modeling, but also you potentially need out of the box thinking, just more efficient policy tools, right? So, some of the other work I have been working on more directly quantify the benefits of the reduced HABs. I have worked with Brent Sundgren, who is at Ohio State Ag Econ Department. Essentially, what we find is we conducted a survey of recreational anglers in 2014. We have found that the anglers are willing to pay $8 to $10 more per trip if they have the option to go through 1 less miles of boating through harmful algal blooms, right? So if you try to quantify and linking that with agricultural conservation policies, that roughly translate into they are willing to pay on average 40 to 60 bucks per acre, per trip, for a policy that reduces phosphorus loadings by 40%. So that shows, that give you some estimates of the potential benefits just for the recreational anglers, right? So we have some ongoing work which look at this even more as well. And one of the message I want to show you is, if you look at the last column using more complex models, that shows you if you reduce the phosphorus by 10%, the economic benefits in total is probably only $8 per trip. If you reduce by 40%, it will be more than 4 times more. So there is a nonlinear effects due to the biological process, and that is also reflected in the economic benefits analysis, right? So some of the work in Iowa, we currently just finished 2 surveys. One is the same questionnaire, one sent to Iowa general public in 2019, and we also sent an augmented survey this year in 2020 to almost 500 Iowa farmers. And what we find is that half of the general public and 30% of the farmers think algal blooms are very harmful, and 60% of the Iowans are at least aware of algal blooms in Iowa's lakes. What is concerning for policymakers and researchers is the red line, red highlighted bullet point, that 65% of the general public are not at all familiar with the Iowa nutrient reduction strategy, which has been in place for about 6-7 years, right? And we have done really good job in educating farmers. So only 20% of the farmers are not familiar with this strategy, but we probably need to up the game for the general public as well. When you are thinking about the policy options, half the public actually chose fertilizer taxes for ag and lawn as the best way to fund this strategy. Well, the farmers' top choice is a recreational fee for parks and lakes, right? The last bullet point in this is that Iowans are willing to pay 19 bucks per household monthly using we use the instrument of the utility bill on average for the combination of 25% less nitrate in source water they are getting, could potentially be groundwater, 50% less HABs, related beach closures, and 10% less Gulf of Mexico hypoxic zone size, right? So those are some of the economic benefits from Iowa. I want to close in by briefly touching on some of the cost of reducing harmful algal blooms and some of the determinants for agricultural conservation practice adoptions. Right. So my colleague and I have done a research that just had a paper accepted by Journal of Soil and Water Conservation. This is based on a special section added to the Iowa Farmland Ownership and Tenure Survey, which we essentially find that in Iowa, 27% of the acres are adopting no-till, and 4% of the acres are adopting cover crops. Recall that in Lake Erie, this percentage is about 20% for cover crops in Western Lake Erie Basin. On the right, it shows you one of the tests that there are things that we also need to pay attention when we talk about conservation by farmers, because half the land in the Midwest are rented out. And when we are looking at the adoption of conservation practices for cover crops, buffer strips, and ponds, we have less adoption on land that is rented out. So this is consistent with this conventional wisdom that when you have land tenure insecurity, you don't necessarily know whether you will rent the land next year or not. You will adopt less investments, including conservation practices. We actually see more adoption on the rented land for no-till, because for a lot of those differences are coming from the landowner who are part-time farmer, so they are renting out on their own ground. They're adopting less. So for the rented out land that is farmed by full-time farmers, they adopt the same rate, right? So again, this points to that land tenure is also an important thing when the policymakers and the extension folks are thinking about who to outreach. Not only producers are relevant, but potentially landowners. We also have some questions for landowners why they are not using cover crops or no-till. For no-till, the reason highlighted are potentially not suitable for the land or potentially hurting crop yields. And for cover crops, and many landowners commented this is a tenant's decision or too costly to terminate or require too much labor or time to plant or terminate. Right? And the top reason for not using buffer strips and ponds are they're not necessarily needed on the land. One of the last few slides that we also have a question asking landowners, what do they think are some of the potential other alternative policies that might induce them to adopt more conservation practices? We ask 3 questions. One is offering you estate tax reduction or exemption when you are for the land that is enrolled in conservation practice, or offer tax-free cost sharing as the second column. The last one and the most, the one that the highest choices, highest percent of the landowner choices is the landowner are really excited about potential additional tax benefits, tax credits for implementation of conservation and practices. And Iowa legislature a year ago, 2 years ago, actually considered whether to offer property tax credits, but it didn't go through. But those are some of the things that potentially has appeal to landowners to further drive conservation practices as well. So the last 2 slides I want to mention is we also have some ongoing work related to thinking about, in addition to cost share programs, what are some of the other policies that we could potentially do? There are some low stakes changes. For example, you can make more induced targeting into the design of the policy rather than paying the same people the same dollar amount, but paying maybe a higher dollar per acre for a high runoff sub-watershed, right? So that's the targeting. Another policy we looked at is potentially reverse auction, is you you say that I have $1 million to pay for conservation practices in this watershed, and as farmers, you need a bid to tell me how much you would need to be paid to adopt a suite of conservation practices. They can choose the conservation practices, but also they bid on the program. That then, based on the potential environmental benefits and the bids, the lowest bids and the bids that can generate the most environmental benefits will be accepted, right? In this ongoing work, based on responses from 500 farmers, we find that when you are using cost share, for example, you have more people potentially enrolled in the program, 25%. When you are using reverse auctions and say that they can bid up to $50 per acre rather than directly paying them $50 per acre in cost share, you reduce the percent of the farmers enrolled to 17 or 18%, but potentially you could increase, you could decrease the cost from the policymaker perspective. You decrease the dollars of the dollar per cost per acre per pound of nitrogen reduced in the watershed, right? Okay. So for the interest of time, I will stop at here. And I want to remind you again that if you're interested about the Iowa Lake Survey, for example, that we'll be releasing the results over the next 2-3 months. Thank you very much. I'm happy to take questions if we have time.
Okay, thank you, Wendong. We got 3 minutes, so let me go through the questions as quick as we can and see what we can do. Did you determine the reduction of phosphorus loading in relation to the increase in cover crops?
So how do we determine that is we worked with a hydrologist who use a SWOT model, soil water assessment tool. So we have a hydrological model which tell us how much loading would generate.
Okay, thank you. Let's do one more. As a farmer, I can tell you that subsidies for CRP do not cover cost of implementation and cost of crop income. Can you comment?
Yes. So, well, so the CRP is a little different. CRP is a set-aside program, but from the perspective of the federal government, even when they're implementing EQIP, they have made a point to not necessarily cover the whole cost. So that's hence the term cost share, right? We do show that when you have higher payments, you definitely can induce more adoptions. But one of the things that EQIP doesn't necessarily have, but CRP has, is there are determination and there are more stringent eligibility requirements for CRP that they essentially look at the erodibility index. So they have some quantification of environmental benefits, right? So including those could potentially yield higher benefits.
What are your suggestions or recommendations for assessing uncertainty in your management policy scenarios?
So what we do is we run simulations in the so we have the economic model, which has a parameter stability. So we run simulations based on that. We also run the outcome based on the wet year and the dry year. When you are looking at the harmful algal blooms, the precipitation, as Grace talked about yesterday, is a major factor, right? So when we are running these, we are taking those into consideration. But that particular paper didn't dive into uncertainty. Okay.
Hey, thank you, Wendong. There are several questions that if you would be willing to type in answers when you gain access to those, be much appreciated. It is 10:25 and we need to move on. Can I ask you to unshare your screen?
Yep. Thank you. I will answer through the Q&A.
Okay, thank you. Our next speaker is Jiyoung Lee. Dr. Lee is a professor in the Division of Environmental Health Sciences, College of Public Health, and at the Department of Food Sciences and Technology, excuse me, at the Ohio State University. She is a co-director of ecology, epidemiology, and population health programs at the Infectious Disease Institute. The main theme of her research is tracking microbial contamination in the environment that leads to human exposure and its linkage to acute and chronic health outcomes. Dr. Lee is currently working on sustainable solutions for cyanobacteria blooms and antibiotic-resistant spread. Her lab is also tracking SARS-CoV-2 infection using wastewater-based epidemiology and linking to COVID-19 trends in cities and dormitories. Dr. Lee, the floor is yours. I will stop sharing my screen, and if you can share yours, it's yours.
Thank you. Can you see the screen?
Yes, I can.
Okay. Thank you very much for this opportunity. I'm extra kind of happy to see Dr. Wendong Zhang and then also Dr. Lesley D'Anglada in the same session. So today's talk is mainly about HAB and human health. And so the outline of today's talk is toxic effects of cyanotoxin exposure using mouse models that we used. So first part will be liver cancer and then gut microbiome disturbance. And then moving to, HAB balloons and then health effects at population level. So we used ecological study design, so focusing on liver disease across the US at the county level. And then the second part will be liver cancer focusing in Ohio at the level of census tract. And then the final question is, okay, the toxins are found in water, and then there are some kind of correlation between harmful algal bloom and some disease. Then what are the exposure pathways? So I'm going to talk about hidden sources of cyanotoxin production and exposures. And then the message will be water is not the only vehicle of our exposure, and the humans are the only ones affected, but also crops and the animals and ecosystems also get affected. So as Dr. D'Anglada mentioned that harmful algal bloom is not just a water problem, this is One Health problems. So I'd like to skip this one because previous speaker talked very well talked about this cyanotoxins, but mostly I'm going to focus on microcystin for today's presentation. So we know that this one is Toledo in Ohio. We have a chronic harmful algal bloom problem. And then the majority of the problem came from toxic cyanobacteria. This picture shows Microsystis, and the Microsystis and other cyanobacteria make toxins. One of them is microcystin, and then this one is Toledo water intake. And then during the severe bloom season, we see this kind of dense cyanobacteria in the water. So water treatment plants do their best to remove all the toxins and then cyanobacteria cells, but unfortunately, several years ago, the toxin was found in the finished water. So what are the adverse effects of microcystins? One is microcystin is known as liver toxin, so it can affect a lot of liver functions, and then chronic exposure can lead to liver cancer eventually. And then also acute exposure, it can irritate a lot of parts of our bodies, including skins and eyes and stomach. And then previous speaker talked about that also, it can affect reproduction system, especially male. Okay, the first part is based on our studies about liver cancer. So we conducted mouse model study, and then looking at chronic toxicity, the outcome was liver cancer. And then we also did acute toxicity, and then another biomarker study, and then gut microbiome. For the chronic exposure to microcystin, we simulated a real-world exposure model through drinking water ingestion. And then we investigate the liver tumor promoting ability of microcystin LR, but also we compare with microcystis extract. So we isolate microcystis from Western Lake Erie, and then one of the toxin producer one, we culture them, extract it, and then we normalized their microcystin concentration. We compare their ability to promote tumors, and then also we help compare the health outcomes between negative control and the positive control, microcystin LR, and the microcystis exposures. So it is a chronic exposure study, and then we used okay, so we used 96 C3H male mice, and then 36 weeks we exposed through the drinking water. So the idea is that the tumor was initiated, and then we added a microcystin or microcystis extract through the drinking water. So the intention was that it reflects high-risk population. And then the result was that when we, at the end of the study, we examined the liver cancer, hepatocellular carcinoma, and then when we compared between microcystin versus lysate, which is microcystin extract, when the toxin concentration was same, then for those exposed to microcystis showed more progressed hepatocellular carcinoma. So you can see this graph that for those microcystic lysis group showed more proportion of carcinoma than microcystin group. So, and then we are quite curious about, okay, then microcystin is ingested or microcystis is ingested. The gut is the first place they got exposed to. And the gut microbiome got a lot of attention in terms of linking to health effects. So he examined their gut microbiome. So and then gut microbiome, also the microbes in the gut have tremendous potential that affect our health, including physiology, metabolic functions, immunity, and even mental health and beyond. And then microcystin and their gut microbiome relationship is relatively unknown, and even less is known about the result of ingesting cyanobacterial matter containing microcystin. Also, gut microbiome, it can be used as a potential biomarker, such as inflammation, before phenotypic impact is shown. So using the same mouse model, so we periodically collect the fecal samples from the mouse. And then this, the major two player was Dr. Sun-Jun Lee and Algal. And then we analyzed their bacterial community using Illumina MySic. The first, we also measured bacteria number between those 4 groups. You can see this graph that for those negative control and then exposed group, about 10 times difference in their bacteria number. So abundance was significantly reduced. And then as you can see, this bacterial culture phenotype is totally different. For those exposed, show much, much less diversity in their group, just when we look at with naked eyes. So we did the bacterial community analysis. As you can see, at the beginning, all the way to the end of this study period, their taxon was changed. So the upper is water, negative control, and then second row is microcystin, third is lysate exposure, and then fourth is positive control fed with known carcinogen. So the microcystin lysate group has much higher ratio of Firmicutes per Bacteroides than negative group. And then also Bacillari and the Clostridia ratio was totally different. Why this ratio is different? I mean, important because high Bacillari Clostridia ratio means they can provide some protection against the inflammatory effects from stress. So at the week of 36, the pattern of bacterial composition in those exposure groups are similar to the carcinogen group. And then this one is PCOA plot. So the number means number 1 is at the beginning, number 5 is week 36. As you can see, when the microcystin lysis exposure progressed, their structural bacterial community in those exposure group were much different from the exposed group. So this color is, yellow color is negative group. So as you can see, exposure group and then cancer group become similar. So the key finding is that bacterial community structure deviate from negative control and then became similar to the carcinogen group. And then the bacterial abundance itself got significantly decreased. And then their gut microbiome disturbance result in more prone to inflammatory effect of stress. And then also they became more prone to metabolic related disease. So, okay, from the mouse study, we found that it can cause negative effects. Then how about population level? What are the public health impact? And then let's talk about liver disease. But first, what is the tool that we can measure the harmful algal bloom coverage and severity? And then from our initial study, we found that phycocyanin is a better predictor of microcystin levels in eutrophic lakes. However, phycocyanin, just the kind of water sample measurement, has limitations. So we moved to satellite remote sensing to capture water color. The study was mostly done by Dr. Stephen Sung, who was my postdoc. So we covered large area of the lakes, including Lake Erie. So we could do multi-year investigation, but also spatial investigation across the US and then Ohio. So we found that we developed the methodology so we could measure severity, and then we tested with Toledo water intake and then compare with laboratory measurements. And then we applied that approach using MERIS satellite imagery, and then across the US, we measure how much area found some detectable blooms. We found that over 63% of the county area covered by detectable blooms. And then we compared to non-alcoholic liver disease, and then we found that there are 65 significant spatial clusters across the US that includes over 400 counties. And then interestingly, the counties in the cluster also showed higher bloom coverage than counties from non-clusters. And then we compared, we did standardized motility rate and then blooms. And then we found that the global association was positive and then significant. And then when we did the model, negative binomial generalized linear model, what we found was that the risk from non-alcoholic liver disease increased by 0.3% with each 1% increase in bloom coverage in the affected county. So then it motivated us to more focusing on liver cancer. So cyanobacteria blooms and then liver cancer in Ohio. So first we used Mary's satellite remote sensing and then found how many areas and what are the bloom coverage, what is the intensity. And then we linked malignant tumor data in Ohio over 15-year data. And then we did census tract level hepatocellular carcinoma incident rates. So we compared those area bloom impacted the surface water for drinking water source, and those area no bloom or those area they use groundwater for drinking water source. So this that part was mostly done by Tyler Gorham. He was my doctoral student, and then through the collaboration with Dr. Elizabeth Root. And then we also used the same negative binomial generalized linear model. And then the hypothesis was that bloom affected surface water, for those population living in the area used bloom affected surface water, probably correlation with increased hepatocellular carcinoma. And then if they live close to the bloom affected area, they have increased liver cancer. So this map shows drinking water source classification in the state of Ohio, but because of the Lake Erie, smaller inland lakes are really small, you can see. However, the color shows cyanobacteria bloom severity based on our satellite remote sensing. And then the black triangle shows non-impacted area, their surface water intake. And then the red triangle means they are bloom affected. And then the public water, drinking water source at the census tract level, the blue color is groundwater, is their major water source. And then green color means their drinking water source is affected bloom. And the white area means no blooms. So when we compared hepatocellular carcinoma incident rate with the blooms between bloom and non-bloom, when they used surface water as their drinking water source, and then non-bloom, the rate is 1, then we compared with the surface water has bloom, we can see that significant increase by 20%, 60%, and then 14%. Another interesting thing is farther from the bloom source has significant decrease. In other words, closer to the bloom, significantly higher hepatocellular incident rate or at the census tract level. So that kind of studies led to next question then, what are the exposure pathways to those toxins? Because drinking water is treated, and then a recreational exposure can be accidental, quite rare. Then why we see that kind of correlation? So we did some study, and that this one is one publication that shows exposure pathways, ingestion, inhalation, and dermal contact. So, and so we were curious about that. Water is the one, how about food? And then we found that food can be an important source of cyanotoxin exposure. So we can easily think of how about the fish caught from the bloom affected area. So we tested 3 types of fish from Lake Erie, walleye, white perch, and yellow perch. This was done mostly by David toward his master thesis, and then also collaboration with Dr. J. Martin and then Dr. Lutson. And then, so we compared those 3 types of fish, and then we found that from their edible portion of the tissue, we found toxins were found at various levels. And then another interesting thing is that a white perch, especially microcystin level, was dependent on local bloom conditions. And then when we did risk assessment, sorry about the BG BG table. So the thing is that microcystin in fish under safe threshold levels when measured with seasonal total daily intake for all population except tribal member or high fish consumers. However, hazard exists for frequent fish eaters and lake anglers when measured with lifetime threshold. So we know that fish can be, or water, but recreational, those can be exposure. But how about irrigation water and drinking water treatment residual? So let's talk about that. So we simulated, like irrigation is contaminated, and then what happened? We use contaminated water for growing crops. So we simulated that situation using MCLR. And then we did lettuce and then carrot, but as Dr. D'Anglada mentioned that root plants, it accumulate more microcystin. So this one shows different part of the crop and then different methods of irrigation. And we used between 1 to 10 ppb as an irrigation. So as you can see, the more toxin in the irrigation water, more toxin accumulated in the crops, but also some portion goes to soil. And then, so the question is, and then if there's a risk, so we did risk measurement and then divide into children and adult population. So when the microcystin is 1, 5, 10 ppb, and then the this is the potential health risk when we compare it with US EPA reference dose and WHO total daily intake. So all the this toxin level show that there is a moderate or high risk based on this food consumption. Then this is an interesting photo we harvest from our study, and then you can see that up until 5 ppb, there is no like a visible difference, but at the time, at the level of 10 ppb, also crops got their quality and yield got also affected. So their length, mass, and diameter, when we compared, there was a significant difference. So crop yield also negatively affected. However, how about other crops? So we tested with green bean, as you can see, as toxin level goes up, their crop quality got really negatively affected, also showing their length, mass, and number of beans also got negatively significantly affected. So it's a beyond human health issue. And then another question is, okay, the bloom affected water for drinking water source, water is treated by the treatment processes, and then where does the toxin and then cyanobacteria goes from the drinking water plants? So those go to land. So from the water, and then now it goes to the soil. And then what happened, we use that one in agricultural land. And then toxin can be accumulated in the crops, or there's a possibility that toxin can be leached to groundwater. So that was the research question, so we investigated. And then this is the one we used a similar approach, and then this is control, and then this was water treatment residual, and then 20% and 40%. So actually, water treatment residual added visually, carrot shows nice kind of food quality. The question is, where does the toxin go? And then edible part, non-edible part, we extracted toxin and then measured. So majority of, in this case, majority of toxin remains in the soil, and then small portion goes to the carrot. And then so the question is, if the toxin goes to the soil, then what is the leachability to groundwater? And then the next question is, okay, so we see that the toxic cyanobacteria from water goes to the agricultural land. can they colonize on the crops and become a new source of cyanotoxic production? So we simulated and then in the greenhouse. So the idea is that, okay, there's a cyanobacteria bloom, directly that water can be irrigated or airborne that can deposit on the surface of the soil or crops. And then if you look at our crops, such as leafy green, there's a stomata. And then this one is our electron microscope. They can go in there. So inside of the crop tissue, on the surface and then in the inside, can they grow and then can they make toxin production? So we examined crop production and then calculate health risk. So this is another figure that shows this is lettuce and then the green are the ones Microsystems. You can see that they can enter over on the surface. So the question is, then microcystin accumulate in lettuce from the newly colonized Microsystems? Yes, this is to control low level, medium level, and then high level Microsystems were applied. We can see that the more colonized, Microcystin accumulate more in lettuce. So the conclusion is that the newly colonized cyanobacteria become a new source of toxin. So that's why it's not just a water problem. So its agricultural environment should be devoid of toxic cyanobacteria. So in a summary, harmful algal bloom, exposure, and health impact, this is my conclusion. So in the lake, either big or small, it's affected directly from drinking water or recreational, not human, but also crops and animals got affected, including pets. For the water portion, that goes to the drinking water plants, and then the water is cleaned and then processed. So for majority of the water will be safe in drinking water exposure. So pets and then humans and then that kind of water will be toxin free. However, then the drinking water plants, the leftover goes to their lagoon, and then the toxin goes to agriculture environment. So then agriculture environment, the toxin also goes to either soil, humans, and livestock, pets, and then crops. So and then also the bottom of the lakes and then ponds got dredged. Where does the dredge material go? And then it kind of another circle is continued. So my perspective when we look at harmful algal bloom is that it is not local, or it is not local, not only local, regional, it is national and then also global problem. So when we look at solutions and policy, we need to look at in a holistic perspective. Then harmful algal bloom is a true example of convergence research and One Health problem. So thank you, and the majority work done by my students in the lab. This is my email address and website. So I will take questions. Thank you.
Dr. Lee, that is very, very good. It's scary. It's for a soil scientist, it does raise awareness that hey, it's scary. We are at 10:54, and the session is supposed to close at 10:55. We have questions in the Q&A box. Would you be willing to respond to those individually? Probably type in answers to those boxes. At 10:55, which we are very rapidly approaching, because we're essentially there, we'll have a 5-minute break to prep for the facilitated sessions for those that have registered. Those will start at 11 o'clock. I'd like to remind everyone that the next sessions this afternoon will start at 1 o'clock. That will be an overview of animal health and HABs. So with that, I will close this session. Ann, is there anything else that we need to do before we vacate for the facilitated session?
No, I don't think so. Thank you so much to our speakers for their great presentations and for helping to field questions as well.
Yeah, they were on target, just absolutely perfect. So anyway, thank you, everybody. Have a safe day, and we'll see everybody back at 1 o'clock.